sample, typically an animal sample. And then from there, we allow that energy to hit the X-ray detector. And so the difference in energy being sent, the attenuation will vary. And so the detector will pick up the signal. We then use various equations and we solve for which, I guess, type of tissue each pixel falls into. So initially it will determine if it's bone or soft tissue. And then that equation is solved again to see if the soft tissue is either lean mass or fat mass. So the outputs from doing DEXA is either a bone signal or fat mass or lean mass. And I'll show you some examples of that as we go through. We also get images using the iNSiGHT system. So you can see on the left-hand side, the X-ray attenuated image, which you would initially pop up on the screen. The system then uses the different equations that we were talking about to determine if the pixel is either bone, which you can see here, or fat or lean mass. So again, the fat is going to be orange and the lean mass will show up as green. We can then do some quantification on those images. So we can draw different regions of interest, which I'll show you on the software in real time, or we can look at the whole animal. And so from either an individual region of interest or on the whole animal, we can get all of these different measurements. So bone mineral content or bone mineral density, as well as the area that's determined to be bone based again on the attenuation of those X-rays. We can then also take a look at fat mass and lean mass, both as a percent and a weight as well. Okay. The iNSiGHT system itself, we'll talk a little bit about the different components of the system, the features and benefits, the technical specifications, as well as the software, and then we'll jump in real time to seeing the system in action. So this is what the system looks like itself. You can see that there's a window on the front that we'll see is very important for being able to monitor the animal while you're doing the imaging, the tray on which the animal sits, and it does also have integrated anesthesia for isoflurane if you choose to use that method of anesthesia. The cabinet itself is self-shielded, so you don't need any other infrastructure to sight the instrument, and so users simply close the door and it's safe to operate. As I mentioned, there is integrated anesthesia if you choose to use isoflurane, and it does only require a standard electrical connection. The software itself, as you'll see when I share it, operates on a Windows 10 operating platform, and there is an offline analysis package available for you to do the analysis on the images outside of the lab, not taking up time on the system to do the analysis and allowing you to sit at your desk or at home to do the analysis if you choose to. The benefit of DEXA imaging and specifically the iNSiGHT system is that it is non-invasive. So you'll see that very quickly we can take an animal, anesthetize it, take the image in less than 25 seconds, and then return it to its cage. There's no preparation steps other than basic light anesthesia required to take the images, and the dose of radiation is quite minimal, so it doesn't have an effect on the longitudinal study. As I mentioned earlier, the scan times are about 25 seconds per scan, and we will see that happen in real time, so you can yourself see how quick it is to get the images and the data that we'll be talking about. The images, as you saw, are quite high resolution, so about 100 microns. This allows us to look at body composition changes in small animals, and the system is very specifically suited for mice and rats, up to about 500 grams. Being able to scan animals that large, we do need a wide scan area. So the scan area is about 16.5 by 25.5 centimeters, again, allowing us to get up to about 500 grams. We talk about rats, but we can talk about similar sized animals as well because it is not species dependent. So at this point, what we're going to do is I'm going to have my colleague Sonika turn on the camera so you can see the system in action. We've spotlighted that for you, so it should show up big on your screen at this point. So what you're seeing now is myself, and you can see the system as well. So the system is composed of the device that you see here, and then the computer and the workstation where the software will be displayed. There is the option for integrating anesthesia, and so this group has a mobile stand off to the side. You could have a stationary system set on top of the instrument as well. It's completely up to you. We will show everything without the animals in view right now. I will use my friendly little mouse here to show how the animal setup works. And then once we get the animals out, we'll turn the cameras off, but I will be able to share the software on the screen at that point in time. So at this point, I'm going to open up the iNSiGHT software. So we see that here. The first thing that you need to do once you turn the system on and start your imaging for the day is to run the daily test. Okay, so I'm going to run the daily test. These are all of the test samples from before. I'm going to click Start Daily Test, and it will show me exactly how I need to position the sample inside the magnet. This is a phantom that comes with the system itself. So I'm going to take the phantom, open the door. It says that I need to place it with the label forward, right in the middle, as you can see there on the X. We'll close the door, and I will hit OK that I've done so and start the daily test. Okay, sorry. Okay, so with the yellow light here, that means that the system is sending out an X-ray signal. There is also an indication on the system itself and an emergency stop should you need to stop the analysis. Okay, so once everything's been processed, we will get a pop-up screen that says daily test passed. It's sending now the second dose of X-ray, so one was high and one is low. You can see the settings that are used here. These are fixed for the DEXA setup. There is the option for digital radiography on the system, which would allow you to change all of those settings and acquire a very specific image to meet your needs. Here we can see that the daily test was a success. We'll click OK, and then we'll take the sample out. The next thing that we want to do is start setting everything up for the animals. And so with that, we have the integrated anesthesia. For the daily test, I put the nose cone up and out of the way so that it doesn't interfere with that test, but you'll see now what we do to incorporate it into the images, but yet not have it interfere with our measurements. So I'm just going to position the nose cone on the platform here. Okay, so that should allow us to have the mouse right in the center of the imaging field of view. I'm going to close the system. So what I want to do now is go into our measurements. Okay, so I'm going to go into the animal list. I'm going to open up our iNSiGHT demo folder, and I'm going to set up a new animal that we're going to do today. So all of these are animals that we've done in the past. I'm going to click on Add. And so today we'll do mouse 1. But again, you can choose how to organize this. So you saw that initial set was just the demos that we've been doing here on this instrument, but you could set up an individual folder for each study that you're doing. And then within that, you would set up all of the different animals that you're going to scan. And so you can scan each animal multiple times and it will assimilate all that data. And I'll show you how we can look at the analysis of historical data as well. Once you're setting up each animal, you put in the study name and the information about the animal. You can enter the sex of the animal, so female or male. You can enter any information you'd like about that animal as well, and click OK. So once we have our animal set up, I'm going to double click on it, and this will allow us to me to enter into the measurement tab. Before I do that, because I have that nose cone in place, I want to let the system know that there's going to be a nose cone and that I'd like to remove it from any of the analysis. So by clicking on Apply Mask, it says this is the old setup that we have for the nose cone. I want to update that because I've moved the nose cone since the last time. Okay, we can see that the system is taking a measurement right now in an image, and that's basically just to identify where that nose cone is, and it will apply a mask to remove that from any of our future images. Okay, so now I can hit apply, and now we have our mask in place. So what I would do now, and what we'll do once we turn the cameras off, is I would have the animal, I would anesthetize the animal, typically using isoflurane. That will be done in a fume hood behind us. Once the animal is anesthetized, we would turn on the isoflurane to the vaporizer going into the instrument. If you get a vaporizer with the instrument, there would be 2 channels, one to allow you to have the knockdown chamber and one then to send it to the system. The way we have it set up here, they're separate vaporizers. So we turn on the isoflurane, I get my animal ready, and I'll have my colleague help me with that. And then we'll put the animal right into the nose cone, apply eye lubricant if required by your protocol. Once the animal is positioned well, we're sure that he's comfortable and not going to wake up, we'll close the door. But we can also monitor the animal through that window, which will allow us to see if the animal's waking up. If we're in the middle of taking a measurement, I can hit that emergency stop and get the animal out without any issue. Once the animal's in place, I will hit measure start, and that will allow us to acquire the image on the animal, and everything will pop up on the screen. But at this point, we're going to turn off the camera. I'm going to share my screen live with you. Okay, and once the camera goes off, I'll have Sebastian anesthetize the animal in the knockdown chamber behind us. So I will talk you through everything that he's doing just so you can understand how fast these images can be taken. I'm taking the animal out, the stuffed mouse out of the system, just so that we have that ready to go. So the animal is just going into the knockdown chamber. And normally, as with everything, it will take a few seconds for the animal to be anesthetized, again, using isoflurane. At this point, it's a great time for you to enter any questions in the Q&A dialog box that you may have. I'd be happy to address them for you. Okay, so we're knocking down the animal. And again, once that's in place, we will turn on our vaporizer going to the system and anesthetize or move the animal over once he's comfortably asleep. Perfect. Okay. So there is a question, the resolution of the system for sure. So the resolution is about 100 microns for the X-ray image. And then you'll see as I go through that we can actually magnify the image to allow us to more precisely place the different regions of interest. And I'll go through that analysis on the animal that we take the picture of. Another question is how many ROIs can be applied? So with a new update to the software, there is unlimited number of ROIs that can be applied. So previously it was 7, but now with the scroll bar that's been implemented, you can apply as many as you'd like. You'll also see that we can apply exclusion ROIs. So if there's something like an ear tag, or you forget to do the mask, you can apply the exclusion ROI to your data to remove anything that you don't want to influence the measurements which are being made. So another question is, can you see a tumor and measure the size of it? So you would be able to see a tumor on the X-ray as long as there was enough difference between the tumor and the tissue around it. On the system itself, the ROIs are, I have to look in to see if you can measure it on there. You could export the image as a TIFF or a JPEG and then import it into other third-party software and do the measurements there. They are 2-dimensional X-ray images, so it would be a 2-dimensional measurement that you're getting based on the size. So another question on can you measure the area and the size of the pancreas of the mouse? I'm going to follow up separately with you on that one, Hui, just because the organ, I'm not sure that it's going to show up well enough on the X-ray to be able to see it, but there's other ways that we can do that with a different system other than the DEXA system. So right now, Sebastian is just moving the animal into the DEXA system. So he's placing the animal on the nose cone, putting eye lubricant on as we go. I'm just going to close the door of the instrument and I'm going to push measure start. So as I do so, the image, the system is going to start to take an image. We know it's sending an X-ray because our yellow indicator is on here as well as on the system. So we have our initial image, and then it's going to send the second X-ray beam through as well. Okay, and that one's done. I hear a tone in my ear as it completes, and now our measurement is done, and Sebastian can take the animal and return it to its cage. So you can see just how fast we are able to take the images themselves. At this point, if you were doing a study where you had to do high throughput and number of animals, you would just put the next animal down and put it into the system and continue to measure. We're not going to do that today. We're going to go through all of the different measurement capabilities of the system. And then Val, again, is going to join us in a few moments to discuss some of the data that he's gotten with the system. So the first thing that you can see is that the nose cone is actually colored purple. This is the color of the exclusion ROI. So we know that that mask is not going to be included in any measurements that we do make. If I want to see very quickly the measurements on the whole animal himself, I just click on the ROI button twice, and you'll see that that brings up the measurements for that individual image. So we can see the bone mineral density, the bone mineral content, the bone area, the tissue area measured, and then the differentiation into fat and lean mass. I'm looking right now at the X-ray image, but I can switch to the bone mineral density. So this is all the area that is considered bone. And then I can switch to the color, which will show me lean and fat mass. So this was quite a small female or male. I'm just asking the team. They're going to let me know. Female? Great. So quite a small female. We can take a look at where that tissue is, and Val will show you some results over a longitudinal study, as well as some coefficients of variation in the measurements that are made. So back here, if I want to magnify the image to draw my different regions of interest, I would simply draw a box around the area that I'd like to magnify and click OK. And so here now you can see with much more detail the different structures that we're seeing. And this is where being able to see the pancreas, I don't think is going to be possible using the system, but something like ultrasound, which is something that I'm happy to chat further about, would absolutely be capable of doing that. If there was a tumor, we'd have to be able to see the difference between it and the surrounding soft tissue. Again, there's other modalities that would allow you perhaps better to do that, but it may be possible with the system as well. If I want to draw different regions of interest, I'm going to click on the region of interest button. Right now, I'm able to draw different areas. So I'm simply going to left click to draw my region of interest. I can draw a few. So this is just an example of taking a look at the femur. If you were looking at bone mineral content and changes due to something like osteoporosis, something like that, you can see the individual vertebrae. You could draw regions of interest there. If you were looking for changes in lean mass or something in a specific muscle group, for example, in the hind limb, you could also do that. If, for example, there was a region of interest that you wanted to exclude, so like that mask, if you wanted to exclude something like an ear tag, for example, I can draw a region of interest around that. And again, as many as I would like. I can also make linear measurements. So if I come and click on, Sebastian, remind me where linear is. Okay, so I'm going to close that one. Perfect. And then the linear measurement here. So if I wanted to make a linear measurement on the femur, for example, I can do so. And we can see, nope, I'm drawing multiple linear measurements there. So you can draw as many linear measurements as you want, different regions of interest. If I want to see the results on those regions of interest, for example, on the femur that I drew, I would just click off of the ROI and you can see them labeled here, and then the results in those specific areas as well. Okay, exporting the data is quite simple. So you can take the data, you can export it as an image or the data itself. So by doing Control-Shift-E, I can export again as CSV or text, and that will be able to be opened in Excel as an example. And if I do Control-Shift-I, the images are going to come out in various formats as well. Okay. Now, if we want to go back and we want to look at that same animal over time. So I'm going to go back to the list and I'm going to go back here and I'm going to go to the Fajardo lab, and the password here is here. And so what we can do is we can go back and we can start to take a look at some history of a specific animal. So this one is one that they did for some of the coefficient of variation testing. And you can see that there's 6 different measurements. And we can see how those measurements compare over time. So here we're looking at the bone mineral density, and here we're looking at the fat percentage. And you can see the individual measurements again over time. If there were multiple regions of interest, you could cycle through those as well. So you could see how those animals were changing within the software, but then of course exporting that data out within Excel to be able to plot and do your statistical analysis and accumulate the data, as you'll see quickly with Val. So at this point, I'm probably going to get Val to turn on his headset, and I'll bring up his slides again so that he can run through the presentation of his data that he's pulled together. Okay? I can hear you now, Val. Okay. Yep.
Okay, so everyone can hear me okay. As Tanya said, we've had the system here for about 2 months, and really the benefit of the system is how easy it is to use. So we've been able to integrate it really well to many projects. I think there's about 4 or 6 projects that we have just in the small amount of time that we've had this system. I'm only going to show you a few of those projects here, and I should give credit to Sebastian, who you'll see all the data here. That's actually the data that Sebastian collected, and he's become our resident expert on this system. So there's 2 main points that I want to talk about today, and that's validation and application. So validation, as Tanya was talking about, thank you. I'm going to be showing you some coefficient of variation. So what we've done here is just repeatedly measure the same mouse with and without repositioning. And we've done it with a mouse as well as a rat. So that may be of interest to some of you here today. And then in terms of application, as I mentioned, we have about 4 to 6 projects ongoing with this small animal DEXA, and I just picked a few to show you today. So our low dose lithium supplementation. So that's actually an update from last webinar. So I'm going to show you some of the longitudinal body composition analysis. And then we have a project with Duchenne muscular dystrophy, and we use the DBA2J MDX mouse, which is a mouse model for Duchenne muscular dystrophy. And we're really interested in measuring the body composition in terms of lean mass, but also bone mineral density and bone mineral content. And then I'll save perhaps the best for last. We've done some ex vivo mouse femur bone analysis in collaboration with NASA. So they've actually sent us some bone samples from mice that were aboard the International Space Station, or the ISS, for 30 days. So I'll just move right along into the validation. And here are some CGs for a female mouse, in this case, a 10-month-old female mouse. And I'm going to show a bunch of these tables here. So I'll orient you to the tables. On the top, from left to right, we have bone mineral density, bone mineral content, bone area, tissue area, fat percent, fat in grams, lean percent, lean in grams, and total body mass. So all those things that Tanya was talking about in terms of what this system can measure. And so we've repeatedly measured this same mouse 6 times. That's why you can see the number of values says 6 all the way across. And with these repeated measures, we can generate a mean, a standard deviation, a standard error of the mean, but of course, a coefficient of variation. And this is what we were particularly interested in, just to see how robust this system would be. And we're pretty impressed with the numbers, all below 3% here in the bone mineral density and bone mineral content. Really impressive numbers for the fat and lean composition, all below 1%. So again, this is in a female mouse, 10 months of age. So then we repeated this in a male mouse, about a year old. Again, same table layout, 6 measures: bone mineral density, bone mineral content, bone area, tissue area, fat percent, fat grams, and so on and so forth. If you look at the coefficient of variation, the last row of this table again, the numbers again are really impressive, less than 3% here for bone mineral density and bone mineral content, less than 1% for the fat and lean percentage and weight. So we're really happy with the performance of this system so far. The next thing we wanted to test was how robust the system would be if we had repositioned the mouse. So again, we repeatedly measured the same mouse. In this case, it's a male mouse. But before each scan, what Sebastian had done was actually picked up the mouse and repositioned the mouse onto the image area, and then clicked measure before each run. And so we can see here that the CVs, again, are really impressive, at or below 3% for bone mineral density and bone mineral content. And below 2%, one just at 1.18, which is still okay with us, with the fat percent, the fat gram weight, lean percent, and lean gram weight as well. So again, really impressed with the performance of the system, even with the repositioning. And for those of you who work with rats, we actually done these experiments, these validation experiments in male rats, aged 3 to 4 months in this case. So the top table is the non-repositioned CV, and you can see the numbers are really impressive, less than 1% for bone mineral density and bone mineral content, less than 1% for the lean and fat mass as well. Of course, Sebastian, what he did was he repositioned the rat, in this case 7 times, 7 measures here, and really still impressive CVs here with 1.15% in bone mineral density, 1.35% in bone mineral content, and then again, less than 1% in the fat and lean mass composition analysis. So really impressed with the validation aspect of this research. So now let's move on to the application, and I'll start off with the low dose lithium and body composition project. And this is actually a project that we have in collaboration with Dr. Rebecca McPherson. She's an associate professor in the Department of Health Sciences over here on the right. And the whole idea is that we're trying to stimulate energy expenditure to combat obesity. And we have data to suggest that low dose lithium supplementation will increase energy expenditure. And so what we provide the mouse with is a small dose. It's 10 milligrams per kilogram per day. And we've shown that this results in a serum concentration of 0.02 millimolar. And that's really low. And we provide this dose for 6 to 12 weeks. Now, this dose has been shown previously and published in 2010 in Vascular Pharmacology to reduce high-fat diet-induced weight gain, which is really important for us. And when we measure energy expenditure in the Promethean metabolic cages, which Dr. Rebecca McPherson has through CFI funds, we can see an increase in energy expenditure in light and dark phases. So if you look at panel A here, we have time on the horizontal x-axis. On the vertical y-axis, we have the rate of oxygen consumption, which is normalized to body mass. And I'll get back to this in the next couple of slides. So this is indirect calorimetry, using oxygen consumption as an indirect measure of energy expenditure. The control mice that were not fed lithium are in black, and the lithium-fed mice are in red. And what you can see here, during the light and dark cycles, the lithium chloride-fed mice will have increased or elevated energy expenditure throughout both periods, so throughout the whole day. And we can see this statistically. This is an N of 10 versus 12 in the dark, light, and daily phases, with lithium chloride significantly increasing energy expenditure. So this is really important to us. And one of the things that we were wondering was, well, if there's an increase in energy expenditure, well, does this translate to a change in body composition? And here are some data that we've pulled up from the small animal DEXA. We've measured the mouse at 4, 6, and 10 weeks. So one of the highlights of this system is the ease with which you can carry out a non-invasive longitudinal study. So we easily measure these mice at 4 weeks, 6 weeks, and 10 weeks. And because it's a low dose of radiation, there aren't too many ethical concerns there as well, which is really important. And what we could see here, again, the lithium chloride is red and the control is black. And on this particular graph, we're looking at fat percent, and we see a main effect of time. So as that mouse is aging, there's an increase in fat mass, which is opposite to lean mass. As that mouse ages, there's a reduction in lean mass. This is normal. We see this in humans as well. But we see also a main effect for both cases, fat percent and lean mass, a main effect of treatment, suggesting that lithium chloride significantly reduced the fat percentage while increasing the lean percentage. So that's really interesting for us as well. And that change in body composition is really important because, as I said, I'd get back to this. Most people will normalize their energy expenditure data or their indirect calorimetry data to kilogram of body mass. Some may argue that normalizing to fat-free mass is more accurate. And so we are able to do that now because we have this system on hand. So we're able to couple the metabolic cage data with the small animal DEXA data, which is really, really strong for us in our field. It's really applicable. And we can see that despite normalizing to fat-free mass, which is a highly metabolic tissue, we see that energy expenditure is still elevated in the lithium chloride group. Again, the red lines here, we can see this statistically significant in the bar graph shown in B in the dark phase, the light phase, and the 24-hour daily period. So this to us tells us if energy expenditure is still increased relative to fat-free mass, then there might be a cellular mechanism underpinning this. And we've got a few things in the works, and we're excited to say that we'll be submitting this for publication in the next few months, but I'll leave it there for now. The next project that we have is on Duchenne muscular dystrophy. So it's a severe muscle wasting disease caused by the absence of the structural protein named dystrophin. So without dystrophin, our muscles become fragile and weak. This is a histological H&E stain. You can see clear signs of pathology, central nuclei, some immune cell infiltration. So the muscles definitely look weak and damaged. There's no cure for muscular dystrophy, so it's a progressive disorder, and it's primarily affects boys because it's an X-linked genetic recessive disorder. And so the boys will experience ambulant complications. They're typically wheelchair bound by the time they're in their teen years. And because there's no cure, unfortunately, they will live a shortened lifespan of about 30 to 40 years. And so the fact that there's no cure really highlights the need to develop novel therapeutic strategies. And really what we use to study other potential mechanisms or therapeutic strategies are the relevant preclinical mouse models. And in our lab, we studied the D2-MDX mouse, which is a more severe and perhaps more clinically relevant mouse model versus the traditional CPD7-MDX mouse model. And we are using the iNSiGHT small animal DEXA to track body composition longitudinally, again, because it's nice and easy to do that with this system, with and without an intervention. So we're treating the MDX mice with a drug. I don't have that data because we just started the treatment. So what I'll be presenting are the baseline data that Sebastian collected for us a couple of days ago. So here is the body composition data for the D2-MDX mouse. And the first point I want to make is that they have lower body mass compared to their wild type mice, which are otherwise healthy. Now, this is well known, it's highly reproducible. We know that the D2-MDX mice will weigh less than their wild type counterparts. However, what we don't know is the body composition. So this data is relatively new to the field. There's a significant reduction in lean mass. There's a significant reduction, a drastic reduction in fat mass. So this together can contribute to the reduction in body mass. And it makes us think that this reduction in both lean mass and fat mass makes us think that there's this hypermetabolic or catabolic state in this dystrophic mouse model. And sure enough, when we place the mice into the metabolic cages owned by Dr. Rebecca McPherson, we can see that the D2-MDX mice, I've got it separated here in light as panel A. B will be the dark phase, and then C is the total daily 24-hour period. And over on the right here, I'll just focus your attention in the light phase. you can see that the D2 and MDX mice, shown here in red, have significantly elevated energy expenditure compared to its wild type, whereas the C57 MDX mice, there's no difference at all. Again, because that D2 MDX mouse model is more severe than the C57. So we see this in the light, dark, and daily period. So that corresponds well with this reduction in lean and fat mass and this reduction in body mass. The next point I want to make is that muscle and bone are connected. Often what we'll see is that muscle wasting is associated with bone loss. Now, this is really important in the context of Duchenne muscular dystrophy because the most common therapy for Duchenne muscular dystrophy is corticosteroid treatment. However, the long-term use of corticosteroids has been associated with increased risk of osteoporosis. So when we're trying to develop new therapeutic strategies for Duchenne muscular dystrophy, we need to be mindful of any potential impact on bone mineral density and bone mineral content and bone structure. And what we could see here in the D2 MDX mice versus D2 wild type, they're already starting off lower in terms of bone mineral density and bone mineral content. So this is going to be a really important outcome measure for us as we test our drugs on these D2 MDX mice. So the last thing I'm going to talk about is the collaboration that we have with NASA. I have here in the title space flight. If you don't use it, you'll lose it. So up in space without gravity, or when you're exposed to microgravity, the loss of the mechanical load leads to loss of both muscle and bone mass. So we really need to develop these effective countermeasures for long duration space flight. You're probably familiar with all the talk about going to Mars. Sure, that's important, but once we get there, we'll have to be functional once we're there, right? So we really need to develop these effective countermeasures that mitigate bone and muscle mass loss. So in collaboration with NASA, and also Dr. Wendy Ward, who's a professor here in the Department of Kinesiology, her and I are trying to target an enzyme called glycogen synthase kinase 3 to determine whether or not that will mitigate the bone and muscle loss that you see with space flight. And what NASA has done, they provided us with some bone and muscle samples from mice that were flown in space for 30 days aboard the International Space Station. And what Sebastian has done has actually scanned the bones, the femur bones in this small animal DEXA system. So again, they are from male mice flown in space for 30 days. And just to orient you to the graphs, we have a ground control. So these ground control, the group they're mice that are housed on Earth in the special NASA cages. Okay, but the bottom line is they're on Earth. The vivarium control are mice that are also housed on Earth, but in your standard laboratory cages. So the ground control is in red, the vivarium control or VIV is in blue. Again, both are on Earth and both experiencing gravity. The flight group, they're housed in these cages, but they're also aboard the International Space Station, in this case for 30 days. And what we can see is that bone mineral content and bone mineral density are significantly reduced in the flight group compared to both ground control and the vivarium control. Now, you may be wondering about the coefficient of variation, of course, because we are interested in the validation aspect of this work as well. So Sebastian has actually repeatedly measured a control bone, and our CVs are still pretty good with just a femur in the system, 3% for bone mineral content and 1.8% for bone mineral density. So this is a really nice finding for us, of course, as we expected, but it's really nice to be able to have this data on hand. The next thing that we want to determine is whether or not glycogen synthase kinase 3 is involved in both the bone and muscle loss. So we'll be processing these femurs to look at GSK3 signaling. But I'll leave it there for now and happy to answer any questions.
Perfect. So at this point, we'd like to launch the audience poll and get a little bit more information about if you want additional information about the system. So there's a few different options here. So happy to reach out and talk more about the research needs that you may have. Additional information about the system. I'm happy to share that information with you. If you're only here to learn about new and interesting technologies, that's okay. It's fantastic for us to know and understand that. And we'll give everyone just a few minutes to reply here. If you filled in the answers to the questions and you have questions yourself, please put them in the Q&A dialog box, because we do have about 15 minutes time, and we can work through those questions between Val and myself, either about the system itself or some of the data that he just presented. And I'd like to thank Val for that time, for his presentation as well. So we'll close the poll at this point and open it up for any questions that you may have. Okay. So you'll see on the screen, there's contact information there. If in the days and weeks to come, you have questions and you want to reach out, please feel free to reach out to us at info@syntica.com. If you'd like to just take a look at the website, feel free to use syntica.com as well. There is one question, Val, for yourself. Does the changes that you see in the bone and perhaps in the muscle, do they recover after flight? And is this something that that is seen just shortly after flight and then it recovers, or do you know much about the time frame of that process?
Yes, they will recover after flight. Most of what I know for recovery is based on, in mice at least, in the earth-based analog, which is hind limb suspension. And you'll see bone and muscle recovery starting at about 3 days, and then will be almost fully restored by 1 week or 2 weeks. Good question.
Perfect. I had a question as you were talking about the cages. It was interesting to me that you had kind of the NASA cage controls on Earth and then the vivarium cage controls on Earth. Was there suspected to be a difference between those, or that was just kind of extra control to make sure there was no effect of those cages?
Just an extra control. We don't really ever expect a difference from ground control and vivarium control, but NASA wants to control everything, right? So, which makes sense.
Of course, of course. I just wasn't sure if there was any altered gravity in the NASA cages or if they were just really standard.
They're really neat, these cages. They have wired caging all the way around so that as the mice are floating, they're able to grab onto something on each side.
Right.
Right. And then their food and is in a special location. So all these things might change animal behavior, right? Which might change your physiological outcome. So that's what we're really controlling for.
That makes total sense. Interesting. How you have to think through all those things that with the lack of gravity, that they're going to naturally want to grasp things in whatever orientation they end up in space. Huh. That's really cool. Sorry, nerding out on science for a minute there. Any other questions, please, again, feel free to use the chat or the Q&A box. I'm happy to walk through them with Val as well. And again, if in the days and weeks to come you have questions, please feel free to reach out. There are a couple of the questions regarding the tumors in the pancreas that I will reach out to each of you directly, just to chat through different ways that may be accomplished. And there's different things that we can do to test that out as well. So thank you for your time. Thank you, Val, for having the system in the lab, and Sebastian and team for working on the instrument and helping Val to pull together the data that he presented. I know I found it really exciting to learn about the different work that's being done here. Hopefully you did as well. And hopefully you found the information in the system demonstration relevant to the work that you're doing. Thank you for joining us today. And if there's no further questions, we'll sign off and give everyone back about 10 minutes in their day, which I'm sure everyone appreciates. But again, if you have questions, again, feel free to reach out. Happy to address them as we end off the session today. Thanks so much and take care.